and welcome to our video. Have you guys ever heard of microbes? In this video, we're gonna talk about bacteria, specifically E. coli. What is E. coli? Escherichia coli, also known as E. coli, is a type of gram-negative bacteria that lives in warm-blooded animal intestine, including humans. Most of these bacteria are harmless. However, certain strains may cause pathogenic reaction which can lead to various diseases. These E. coli are gram-negative bacteria. They appear pale reddish color when it is observed under microscope. This is due to the thinner peptidoglycan layer in gram-negative cells that allow the crystal violet to wash out in addition of ethanol. In 1885, Dr. Theodor Escherich, a German-Austrian pediatrician, discovered E. coli on the specimen of feces from breastfed infants.
Most prevalent reproduction of E. coli is asexual reproduction, which is binary fission. Fission of chromosomes shows the beginning of this process and followed by the cell elongations. Then, the cell membranes start to pinch inwards and forming the transverse wall. The septum form and leaving the two daughter cells at the end of this process. There is no transfer of genetic material in this process, but the cell divisions do occur. Commonly, E. coli undergo genetic recombination through conjugation pillars, which this process involves the transferring of genetic materials between two bacterial cells through sex pillars. It begins with the E. coli that has a plasmid, or also called the donor cell. The donor cell and the recipient cell will be linked together and forming the conjugation tube or pillars. The donor cell have the F factor, which make up the sex pillars. After finish replicating its own genome, the donor cell will start to transfer the DNA molecules to the recipient cell in the form of plasmid. Furthermore, the recipient cell will continue this process by duplicating the transferred DNA. Finally, both of the cells will move apart and leaving the two circular plasmid in each of them. The F factor that has been transferred into the recipient cell turns it into the new F plus donor.
E. coli is the best or most studied free living organism and has recorded 11 prestigious Nobel Prizes associated with it. More than 700 serotypes of E. coli have been identified. Most of E. coli not only live in the intestine of a healthy human being, but also helps to keep its host healthy by helping digestion and even defending its host against harmful microbes. Most of the variety of this microbe is benign, so it makes the bacterium an excellent lab pet. You can do experiments without a lot of sophistication. In addition, it is growing very fast. A single bacterium placed on a nutritious jelly can form more than 1 million of its own in 24 hours.
So I'm going to present the applications of E. coli. In addition, E. coli plasticity and tools such as biobrick building can facilitate plasmid and reporter constructs. Correlations of cell growth and physiology with expression patterns from reporter constructs can also help to expand our knowledge of the exposure impact to the external stimulus on cell physiology. Biosensors based on whole cells are inexpensive alternative and can be incorporated to portable devices. Using qualitative reporters, we can apply it in field research. One good example is the detection of parasites without using cold protected samples or complicated equipment for the detection process.
Symptoms of Shiga toxin producing E. coli varies in individuals. Some of the symptoms occur within 3 or 4 days after the exposure, while others may become ill as soon as a day or a week later. They include severe stomach cramps, diarrhea, which is often bloody, vomiting in some people, while severe cases may include bloody urine, decreased urine output, pale skin, and dehydration. Causes of E. coli is because of improper food handling. First is because of failing to wash hands before cooking or eating food. Second, consuming foods that is contaminated with E. coli bacteria. The food is not cooked properly. And last but not least, drinking contaminated water. Always wash hands after using the toilet. Cook food properly to ensure all the bacteria are killed. Wash all products before eating and avoid leaving food outside and refrigerate leftovers immediately. Conclusion: E. coli is versatile, adaptive, and have the ability to communicate and transfer DNA.